Hello and welcome everyone to this podcast for human genetics. In this podcast, we're going to start our discussion on what your book calls Beyond Mendel's Laws. Now, to introduce this idea, it's important to remember that when Mendel's studies were rediscovered in 1900, several decades after he had died, many scientists were trying to see if they could get the same ratios that he predicted in different organisms. And what was discovered was that most often researchers found consistent results. However, every now and then they discovered that genetics was not quite as simple as we may have originally thought after Mendel's studies. In fact, many traits today we now know don't follow his predictions perfectly. But let me explain that. So remember, with Mendel's studies and his laws, he would predicted that if you took two heterozygotes, say big A little a times big A little a, and you crossed them, and we could do a little Punnett square here. The prediction for the genotype would have been a 1 to 2 to 1. That is 1 big A, one homozygous dominant, 2 big A, two heterozygotes, to 1 homozygous recessive, little a. And remember, this would have been the genotypic ratio, or the genotype ratio. But let's not forget about our phenotype ratio. So let's write that next. And from Mendel's laws, we would have predicted a 3 to 1 ratio. That is, 3 would have the phenotype of big A, and 1 would have the phenotype of little a. Now, it turns out that when they started seeing these alterations of Mendel's predictions, it wasn't because of the genotypic ratios. In fact, the genotypic ratios remained consistent. Regardless of what the differences are that we're going to see the phenotype ratios, remember that the 1 to 2 to 1 prediction of Mendel's laws remained constant. But what was discovered was that there were some variations of the phenotype ratios. Maybe they weren't always 3 to 1. Maybe sometimes they were 1 to 2 to 1, where some of these maybe didn't behave as Mendel would have predicted. There were some alterations of the phenotypic ratio that showed a 2 to 1 segregation. Prior to Mendel doing these studies, we didn't know about sex chromosomes, so sex chromosomes added some complications. We also know that the environment plays a role. So what we're going to do for the rest of this podcast, and it might extend into a second podcast, is talk about 12 different extensions to Mendel's laws. So I'm going to list all the extensions first, and then we'll talk about each one individually. The first are lethal alleles. And when we talk about lethal alleles, the example we're going to focus on is achondroplasia. We're also going to talk about something called multiple alleles. And for an example of multiple alleles, we'll talk about cystic fibrosis. Our third example will be incomplete dominance. And our example will be hypercholesterolemia. We'll call it HF for hypercholesterolemia. We'll also talk about codominance. And in talking about codominance, we'll talk about the ABO blood types as an example. The next one we'll talk about is overdominance. This is also known as heterozygous advantage. Several interesting examples of overdominance, but the one I'm going to focus on is sickle cell anemia. The next one will be epistasis. And I'll use as an example for epistasis a blood disorder called the Bombay phenotype. 7 and 8 kind of go together, and that's penetrance and expressivity. The example we'll use to describe penetrance and expressivity is polydactyly. The next one will be pleiotropy. And as an example of pleiotropy, we'll talk a little bit about Marfan syndrome. Number 10 will be phenocopy. As an example of phenocopy, I'll talk about deafness. Number 11 is something called genetic heterogeneity. The example for genetic heterogeneity we'll use is blindness. And then our last example will be mitochondrial. For this example, it's a large class of disorders that are just collectively called mitochondrial myopathies. Some of these we'll spend a little bit more time on, and others we'll go through a little bit more quickly. But let's go ahead and jump right into lethal alleles. The phenomenon of lethal alleles was first identified in 1905 by a French scientist of the name Cugnot. I'm not going to ask that on a quiz or an exam, but sometimes people like to know some of the history behind it. Now, Cugnot's organism that he worked with were mice. And the mice that he worked with had a wild type color that we call agouti. It's a funny word, but I'll explain what that means in a moment. Agouti is a blend of black and yellow. When you look at them, they look more black. Now, they also had a mutant mouse color that was yellow. So what Cugnot did next was he took the wild type agouti mouse and he crossed it with the mutant yellow mouse. What he got was an approximate 1-to-1 ratio of agouti wild type to yellow. So remember, in 1905 they had just uncovered Mendel's work, so they assumed then that yellow was dominant. So they might have predicted that the genotypes of these two would have been something like this. For the wild type agouti, it might have been little a, and then for the yellow, it might have been big A, little a. So next he wanted to prove that the allele that causes yellow was dominant. And so what he did was he took two yellow mice and he crossed them. Now, if big A is dominant, what you would see is this: big A, big A, big A, little a, big A, little a, little a, little a. If big A, in this case, is dominant and causes yellow, so if that is true, after he did this experiment, he would have predicted 3 yellow mice to 1 agouti colored mouse. What he discovered was a 2 yellow mouse to 1 agouti mouse ratio. And the reasoning for that, we now know, is that this mouse is yellow, this mouse is yellow, so let's just write that here. Big A, little a is indeed yellow, just as they predicted. And little a, this one here, is agouti. So what about big A, big A? Well, it turns out that in the combination of big A alleles, that is lethal. Big A mice die in utero. So this is a lethal allele. So let's give it an actual definition. Lethal alleles are allele combinations that cause death. In reality, a lot of allele combinations can lead to death. In fact, a lot of diseases that are recessive would be considered lethal alleles. Cystic fibrosis is considered a lethal allele because people with that disease, little c, for instance, would eventually die due to the symptoms of cystic fibrosis. But what I want to use as an example today is a disease called achondroplasia. If you're a fan of Game of Thrones, the actor who played Tyrion, Peter Dinklage, has this syndrome. And similarly, as the mice that we just talked about, there are some combinations of alleles that lead to achondroplasia, and some that lead to death, and some that lead to an individual without this syndrome. Individuals with achondroplasia typically have a relatively long trunk, but short limbs, arms and legs. They will also typically have a large head with a flatter face. So let's look at it from a genetic explanation. Somebody who is achondroplasia will be heterozygous, and I'm just going to use A for simplicity. So they might be big A, little a, where A, the big A allele here, is dominant over the little a, which leads to the syndrome of achondroplasia. If two people with achondroplasia were to have children, the progeny of these two achondroplasia parents would have genotypes of big A, big A, big A, little a, big A, little a, little a, little a. This person here, little a, would not have the syndrome. They would be of average height. Those who are big A, little a, either this one or this one, would have achondroplasia. Individuals who are big A, die before birth. This combination of big A is a lethal combination. For the purpose of this class, if I talk about an example which may not be achondroplasia, but any example I give on an exam or a quiz, and the result is a 2-to-1 ratio, there's a good chance I'm talking about lethal alleles. Now I'm going to come back over here and look at this and give Mendel the props he deserves. Genotypically, the ratios still work out with a 1 to 2 to 1 ratio. It's just that this one genotype, big A, isn't included in our observable phenotypes. So anytime you see a 2-to-1 ratio, you can assume for the purposes of this class that it's a lethal allele example. Okay, the next example I want to talk about is something called multiple alleles. Now, when Mendel did his P crosses, we talked about his crosses involving the round allele and the wrinkle allele, where the genotypes for round would be big R, or big R, little r, and for wrinkle it would be little r, little r. What we now know is that within any given population, there are not just two alleles. In fact, rarely are there just two alleles. There's not usually just, say, in this case, big R and little r. There might be several mutant alleles. So little r1, little r2, little r3, and so forth. There might be other alleles that have a somewhat round wild-type phenotype, say big R1, big R2. There's all kinds of alleles. Now, in some combinations, those alleles can have different phenotypes. For our example of multiple alleles, let's talk about cystic fibrosis. For a long time, we assumed two main alleles. Big C paired with either a little c or big C would equal a healthy individual. And then somebody who was little c would have cystic fibrosis. People with cystic fibrosis typically have severe lung infections, very susceptible to various kinds of bacteria and viruses. Lungs are highly congested, and because of their constant sickness, they're often slow to gain weight, or might just be underweight their whole life. And typically they die at a younger age. Now, we are much better at treating the symptoms of cystic fibrosis, and so now individuals with cystic fibrosis typically live a lot longer than they did, say, 20 or 30 years ago. Now, what we have discovered, though, is not everybody with cystic fibrosis has the same degree of severe lung infections. They're not always highly congested. Some of them gain weight normally. And so what we know is, in the more severe symptoms of cystic fibrosis, and I'm just going to call it little c1, we know this is their allele combination. But we also know that there are other alleles that cause cystic fibrosis, of still the same gene though. say, let's just call it C2, C2. These individuals might not be quite as sick. They might have bronchitis, might have more sinus infections, and other combinations, and we'll just call this C, little c3, little c3, might have relatively healthy lung functions that turn out to be sterile. And this is only seen in males. There's not a connection to female sterility, but certainly with male sterility. So because within any given population we have many different alleles, sometimes in different combinations you'll get different phenotypes. I wrote these as all homozygous little c combinations, but of course you could have a little c1 and a little c2, or a little c2 and a little c3, whichever combinations you might dream up here. And they're going to end up having variations of these symptoms. That's an example of multiple alleles. And again, most traits are associated with multiple alleles. Sometimes we don't know all the different alleles that can lead to various traits, but more often than not, when you look at a particular trait, it's not just because of two alleles. Okay, now, as I just said that, I remembered that there is something in this concept that sometimes trips up students, and I don't want you to be tripped up by this. So let's just back up a little bit, and I want to clarify something. Within a population, there are multiple alleles for any given gene. But don't let that confuse you, because individuals only have two alleles. So a person may be big C, little c1, even though they're living in a population that has a population of humans that have all these different alleles. Big C, little c1, little c2, little c3. Any individual only has two alleles. Next, I want to talk about this concept of dominance. We've already talked about complete dominance. And just to remind you, that's when A is dominant to little a. Big A is dominant to little a. And because of that, somebody who is big A has the same phenotype as somebody who is big A, little a. And thinking about complete dominance, that's why when we do a Punnett square of a heterozygous cross here, is why we see this 3-to-1 ratios, because of complete dominance. Now there are three other types of dominance I'd like to talk about. Incomplete dominance, codominance, and then overdominance. So incomplete dominance is when you have a heterozygote, say big A, little a, that has a phenotype. So it has a phenotype that is a blend of two extremes. So it's a blend of little a to big A, big A. So a classic example of incomplete dominance that you see in many textbooks, genetics textbook, is when you take two pink flowers. And we now know that they're heterozygous. And when they're crossed with each other, what we see are 50% of those progeny will have pink flowers, 25% will have red flowers, big R, and then 25% will have white flowers. I don't have a white marker because white marker on white, of course, would be white. That'd be hard to see. But what we see here, as far as the phenotypic ratio is concerned, is a 1-to-2-to-1 ratio. 1 red to 2 pink to 1 white. As you see here, the genotypic ratios hold up to Mendel's predictions. 1 big R to 2 big R to 1 little r. It's just that the phenotypes come out differently. So now let's think about a human example of incomplete dominance. And the best one to talk about here is something called hypercholesterolemia. Because we don't want to keep saying that, we're just going to call it HF. Someone who has hypercholesterolemia, and we're going to call that little h, they have extremely high cholesterol. levels in their blood. It can be anywhere from 600 to something greater than 1,000. I'm not going to ask you to know these levels on an exam, but you should know that they have deadly levels of cholesterol. Somebody who is big H, should have, under a somewhat normal healthy diet, somewhere between 150 to 200. Now, in reality, a lot of people are slightly above 200, but you would want your cholesterol to be under 200 is what is often suggested by doctors. And at this level here, you have a really low risk of having a heart attack. These individuals here, who are little h, often die in childhood due to heart disease. They will have massive heart attacks as a child because of this extremely high level of cholesterol. Now remember, the definition of incomplete dominance is that the heterozygote, so in that case big H, little h, is going to be a blend of something in between. And so what happens here is that those who are heterozygous have high cholesterol. So let's put high cholesterol. And I don't mean just somebody who's big H and has a poor diet like myself, and it's a little bit over 200. These people have high cholesterol under a somewhat normal diet, and their levels might be anywhere from 300 to 600. So they can take medicine to kind of keep this lower, but these individuals will often have heart disease at a younger age, but they won't die in childhood typically. But they may have a heart attack, say in their early 20s or 30s, because of the cholesterol problem they have. Again, this is a phenotype, the big H, little h, in between the two extremes. If this was a case of complete dominance, the big H, little h would have this phenotype, because in that case, H would be dominant to little h. But that's not what we see here. We see incomplete dominance, a phenotype in between the two extremes. So anytime you see for the phenotype, a 1 to 2 to 1 ratio, where this middle one here, the heterozygote, is a blend between the two extremes, then you know we're talking about incomplete dominance. Let's move to codominance. And so let's just go and cross off incomplete. We're done with that. And let's talk about codominance. The best example of codominance is to talk about blood types. Codominance is when you have different alleles that are both fully observed in a phenotype. So if we think about our example of big A, and little a, and the heterozygote of big A, little a, the phenotype of big A is that you would fully see the A phenotype. Little a, we would fully see the little a phenotype, whatever that might be. We'll talk about the example in a moment. But in the heterozygote, what you would see is this. You would fully see both big A and little a phenotypes. And to better explain this, let's think about blood types. So our ABO blood types are determined by this gene that we call the I gene. The I gene can exist as multiple alleles. It can exist as I with an A superscript, an I with a B superscript, or as a little I. Not only shows us codominance, but it also, as you'll see in a moment, offers an example of complete dominance and as an example of multiple alleles. So multiple alleles because there are 3 different alleles here. So we know that any individual is only going to have 2 of these 3 alleles. So an individual might be AA. So let's remember what I'm showing here is the genotype. And over here will be the phenotype. And so the phenotype of somebody who is AA is blood type A. Now A is completely dominant to little I. So this phenotype would also be type A. Somebody who has the genotype of big B would have the blood type of type B. B is also fully dominant to little I, would be type B. So from just looking at this, we can see that little I is recessive to both A and B. And A and B are both dominant to little I. And if you have two little i's together, like so, that is what is known as type O blood. Now the only combination we haven't talked about yet is AB. If somebody has both the A and B alleles, they have the phenotype of type AB. So if A was dominant to B, the phenotype would be A. If B was dominant to A, the type would be B. But what we see in this heterozygote here, AB, is we see both phenotypes of A and B fully expressed. And so we can't call it A or B. We have to call it type AB because they're both expressed. To review, it's an example of multiple alleles because there's multiple alleles. It's an example of complete dominance because both A and B independently are dominant to little I. And it's an example of codominance because as a heterozygote, A and B have a codominant phenotype of AB. The blood type system is also an example of epistasis, which we'll talk about in a little bit. Overdominance, and I should remind you that sometimes I will interchange that with the term called heterozygous advantage. They both mean the exact same thing. And in this one, I'm going to use the example of sickle cell anemia. Sickle cell anemia is when your red blood cells take on a different form. So a wild type blood cell, red blood cell, will have a disc-like shape, but they're very elastic and they can move around our circulatory system in a very efficient manner. They can stretch and bend and they can make it through our tiny capillaries to get to our organs. Now, somebody with sickle cell anemia, their red blood cells, as you might imagine, instead of being round and elastic, they're sickle shaped. So let me write elastic here. And sickle cell shaped red blood cells are very rigid. You don't have to know why they're rigid. If you ever want to know, we can chat about it. But for the purpose of this class, we don't need to know about that. One of the biggest problems with these sickle shaped red blood cells is that they get stuck in our circulatory system. They get stuck in those capillaries because they're not flexible. They're not elastic. So they get lumped up into our blood vessels and it restricts the flow of blood to vital organs. It can lead to episodes of increased pain because of the swelling, particularly around the joints. It can lead to strokes. It can lead to swelling of the hands and feet. Often individuals could have vision problems. And so what we know is somebody who is homozygous, dominant for the sickle cell gene, has normal shaped red blood cells. And somebody who is homozygous recessive will have these rigid and sickle cell shaped red blood cells. Somebody who is heterozygous will have normal shaped red blood cells. Now sometimes they may also form some sickle cell shape, but in general most of them are of the healthy variety, the elastic disc shaped red blood cell. Now the reason we call this overdominant or heterozygous advantage is because these individuals have an additional benefit. And it turns out people who are heterozygous for sickle cell don't have the anemia, but interestingly, they are protected against the parasite that causes malaria. So that parasite has a life cycle that involves infecting the red blood cell and can lead to sickness and death. Now, interestingly, as we said, the heterozygote is protected against it, and for some reason, the parasite cannot complete its life cycle through a heterozygous red blood cell. And so those individuals are protected. Now, also, people who are little s are also protected against malaria, but it doesn't do them as much good because they have sickle cell anemia. These individuals don't have sickle cell anemia, and they're protected against malaria. If you were to see an example of overdominance on an exam question, what I might have is that as a homozygous, homozygous dominant, the individual is healthy. As a homozygous recessive, they are sick for some reason, and I would explain the sickness. And then as a heterozygous, they are healthy plus some benefit. This helps explain why evolution hasn't simply gotten rid of the little s allele, because you might imagine that since there's such a deleterious effect, a negative effect of being homozygous little s, because you die of sickle cell anemia, why would evolution select to maintain this dangerous allele? And the answer is that it protects a population that is heterozygous for it. We see lots of examples of this within the human genome. Well, I think this is a good place to end this first podcast on the extensions of Mendel's rules. We'll pick up on the next podcast from our list and start with epistasis. If you have any questions at all, please let me know. If not, I'll see you on the next podcast. Bye.